So as Nat said, we've got a few contributors this evening, and not all of them could be with us, but I did especially just want to point out Cecilia Langhorne, who put a lot of work into some of the content from tonight, and there'll also be a short video from Ruth later on. I've got my email address down the bottom left here. If anybody has any questions, if I don't cover anything in enough detail, or if you need to know anything else, I can do my best to answer any questions or pass you on to any of the other contributors from tonight as well. So if you were here a few seminars ago when Leah, Benson and I were talking about gamete assessments, you might recall this figure, and I wanted to show it again just now because I think it's a really good sort of summary of what this webinar series has set out to achieve, and it sort of gives us a good idea of where we are now. And so we started off with like the gamete development and just basic reproduction, the different methods of gamete retrieval, and then we covered the gamete assessments. And then tonight we're going to zoom in on the gamete storage, in particular cryopreservation. So when I sat down to start putting together some of the content that we all had pulled together, I had these sorts of questions in my mind, you know, why do we do this? How do we do this? When do we do this? So hopefully between tonight's seminar and an upcoming seminar in a couple of weeks, you'll get the answers to all of these questions. So we'll start off with the why. Why do we biobank? And this probably isn't new to a lot of you, but I'm going to cover it anyway. So preserving genetic diversity for declining species is one of the main reasons, but also we can achieve better genetic management within breeding programs. And I'll get into that a little bit more closely in a moment. And so one of the golden standards that we think of when we think of this outside of the amphibian realm is the black-footed ferret. And many of you may be familiar with the story behind the black-footed ferret. But in case you're not, this species was thought to be extinct many years ago, and then they rediscovered a small population. They took this small population into captivity, and luckily they were able to cryopreserve sperm from the founder males. And then 20 years later, when assessing the genetic health of the colony, they found that it was becoming quite inbred, and they were actually able to restore the heterozygosity of the population by inseminating females with semen from the founder males that had been cryopreserved. And in fact, they recently had another story that sort of just helps highlight why assisted reproductive technologies are really important for wildlife conservation, because the San Diego Zoo was able to thaw out a frozen cell line that they had from a unique female. So this was not one of the founder genes or hadn't contributed to the existing population, and they were able to clone that female. And so it's just a really nice story to highlight why we do these things and what we might be able to achieve 20 years down the road. So as I mentioned, we can also use it for better genetic management, and what this translates to is cost savings. So this is some results from a recent study by Lachlan Howell out of the University of Newcastle. He did some modeling, and what he did for these two frogs, and there's a second paper with a different frog as well. He looked at the cost of running a traditional breeding program, and then he also compared the cost of maintaining 90% heterozygosity by reintroducing founder males at regular periods throughout the breeding program, and found that with introducing cryopreserved sperm from founder males, you could reduce the number of males and things like that. And this resulted in a significant cost savings. So I'm going to go through some key points for cryopreservation, and much of the content here is from Dr. Langhorne. If you have any questions, I'd be happy to put you in touch with her as well. So a few years ago, we reviewed in Clulo et al. The number of cryopreservation protocols that had been published over the years. And at the time that totaled 37 Anuran sperm cryopreservation protocols. And this has increased a little bit in recent years, especially with the special edition from Reproduction, Fertility, Development that Natalie Calatayud piloted, and that was really cool. So I encourage you all, if you haven't, to go and have a look at that. And there's quite a few new papers in there as well for both amphibians and reptiles. So that was a really valuable thing for amphibian cryopreservation and conservation. But prior to 2011, the majority of studies use testicular macerates. And I want to add at this point that testicular macerates, while they're not ideal because it's obviously a lethal process in most cases, it certainly does have its place in conservation. You know, there's things like opportunistic, when sometimes animals might die, but also a lot of museum voucher specimens, stuff like that, have been an opportunity for me to collect testicular sperm in the past. But in 2008, there was an example of non-lethal sperm collection and subsequent cryopreservation, which was quite good. So it's good because since then, there have been more studies turning up where we haven't needed to resort to lethal methods to collect sperm. And I know that Justine, who will be talking, oh, sorry, Rebecca Hobbs is going to talk later, but Justine has a paper, Obringer et al., that has an early example of sperm collection, non-lethal as well. And so another point is that oocytes and embryo cryopreservation has not really been achieved in frogs or salamanders, and this is largely due to the big size of the oocytes and the fact that they have a really high yolk content, which makes these structures really difficult to freeze without ice crystals forming and things like that. But there have been recent developments in the area of corals and fish with basically ultra high rates of freezing and thawing that have allowed some advances in this area. So hopefully in the near future, we might see some of that applied to amphibians. Often cryopreservation protocols can be species specific. And while it's probably unlikely that we'll come up with a cryopreservation protocol that we use across every single species, that's not to say that there isn't some protocols that can't apply to multiple species. And so on that note, often a preliminary investigation using a common related species is recommended. Usually this is if you're using, if your target species is like a super endangered frog or something that you don't have easy access to. So if you're like going into remote field situations, working up your protocol in a common species that is easy to access is certainly something that's very useful. Often we see in conservation a trial and error approach where you sort of get these different individual variables that you manipulate. Maybe you change the thaw rate, maybe you change the cryoprotectant and see how that affects it each time. I think this is often a result of the fact that, you know, in conservation, sometimes these things, I mean, it would be ideal if we had all these protocols worked out years before we needed them and stuff like that, but that's not always the case. So sometimes you sort of get to this last minute where you have this sort of trial and error approach. But of course, if you can do it methodically before you need the protocol, that's great as well. So to that note, if you do find yourself in a situation where you need to figure out a protocol quite quickly, we would recommend reviewing the literature to see if there's a similar species or a more related species that you could use a base protocol for. And of course, naturally, it's hard to publish everything as soon as you know it. And so I would also recommend reaching out on the Slack channel that Natalie's created in case anybody else can help you with your particular species of interest as well. So this is a nice little table summarizing the cryopreservation process and sort of the steps you would go through for a protocol development. I believe this is from a recent review paper by Della Togna in 2020. And so for stage 1, we're sort of talking about, similar to that figure I showed at the start of the seminar, you've got your sperm collection, such as hormone induction, or maybe you're macerating a testis. You would do your sperm evaluation, which is extremely important that you have a baseline wherever possible before you freeze something, so that you can see how successful your protocol is. And then, you know, if you can, you would do sperm morphology and things like that as well, depending on what resources you have and how much sample you have to spare, of course. So then stage 2, which is something that's often neglected and often not reported, but can be extremely important as well, is your cryotoxicity, which is sort of looking at the effect of your cryoprotectant, whether permeating or non-permeating, on a sperm cell without cryopreservation. So many of these things that help preserve the cell during ultra cold temperatures are quite toxic to the cell at room temperature. So it's this catch-22 thing where, for example, in a species that I've worked with, glycerol has a negative effect on the sperm before you even freeze the sperm, which leads to not very good post-thaw results. And so it can be quite useful to have that sort of data before you waste too much sperm in the actual freezing, or if you only have a certain amount of sperm to freeze, ultimately, then it's useful to know this sort of thing. Even if you can only do it in a related species, it's still quite useful. So this would involve, basically, it's a good way to narrow down your protocol for cryopreservation as well. So you would expose your sample to a range of concentrations of the cryoprotectants that you would plan to use, and you would maybe check the effect on, say, motility or cell viability at a range of times after addition of that cryoprotectant. Stage 3 and stage 4, the freezing and thawing and the sperm recovery, I'm going to go into in a little bit more detail. But essentially, you know, the actual aim of the whole talk is to go into, you know, the why, like how to freeze and what you would actually do to freeze. So I'll go into more detail with that now. So with freezing, you generally have an inverse relationship with your cooling rate and concentration. So for example, when you vitrify something with a very fast cooling rate, essentially instantaneous freezing, you would use a very high concentration of CPA, which is your cryoprotectant. Whereas if you were doing the sort of a slow freezing rate, you might use a lower concentration of your cryoprotectant. And sperm samples are typically frozen in straws or cryovials, but it is also possible to do things such as pelleting over dry ice. I think Ruth will talk about that a little bit shortly as well. So it's important to note that all of these things can have an effect on how the freezing and thawing rate should be applied. So for example, with a cryovial, you're using a much larger volume of sperm and cryoprotectant, and so the outside portion will freeze a lot faster than the inside. So that can cause differences in how the sperm have, whether or not they're successfully thawed out. So yeah, I would usually recommend using straws where possible, but there are definitely protocols that utilize a whole bunch of different things. So I'll stress straight away that these are not the only ways to freeze sperm, but these are some of the more commonly used things. And also the seminar in a couple of weeks will get into a bit more of the field-friendly methods as well. But one of the most common things is a programmable slow rate freezer. These are great because they're highly controlled. You can program a whole bunch of things. You can make it multi-step. You can make it just a straight line, like a standard just one rate of freezing from start to finish, or you can do stepwise. They're easy to manipulate, essentially. They can be more expensive than other methods, but it can be useful, especially if you're doing larger quantities of sperm freezing. So something else that a lot of people use would be liquid nitrogen in a styrofoam box. And you can use a data logger with a thermocouple to determine your freezing rate, which is also something I would always recommend, is recording your freezing rate to go along with all the data. I think Rebecca is going to talk a little bit more about data storage and stuff when she has her talk later. And then these things, like I said, can be more portable, so you could use them in the field, but there are certainly other methods you can use in the field that we'll get into in the next seminar in more detail. These are much more cost effective. You need access to liquid nitrogen, and you need a way to suspend your sperm at a fixed level above liquid nitrogen, which is there's quite a few ways to do that. It's important that you document everything you do here very carefully, so that it's all replicable when you write it up in your papers and things like that. So the success of your sperm cryopreservation, and this has been covered in previous seminars. If you've missed those, you can go back and watch them. I believe they're on a YouTube channel. So the way that we determine the success of these protocols is usually by sperm motility and other functional capacities, such as like whether the membrane is still intact or whether they can fertilize, that sort of thing. So it's important that if you have enough sample, that you save some for quality control, or so that you can thaw it out and have a look at how useful that protocol was. For thawing, there are two methods that are most commonly used. Would be fast, often 40 degrees in a water bath, but that could vary from protocol to protocol, and slow, which is usually on a bench top, just at room temperature. And this is something that I have had a lot of success with in the past. I will say that the optimal thawing rate can very much depend on the rate that you froze the sample in the first place. In addition, the way that you thaw might depend on what storage container you used, as I said, what freezing method you used, and also, yeah, you have to consider how you're going to thaw them. So if you're doing it in water, you have to make sure you're not going to lose the sample into the water bath and things like that. And again, it's useful when developing these protocols to consider individual manipulation of each of these variables to figure out which one is having either a positive or negative effect. So as I mentioned already, the most commonly reported things that we do to test the success of these protocols is post-thaw sperm parameters such as motility, and often sperm viability, being membrane integrity. But there are some reports, and there are more and more reports every year. Like, this is increasing, which is great. There's way more sort of concept of proof of concept, rather, that there are able to be fertilization and survival from larval through to metamorphosis in many species, which, you know, the ultimate indicator of our success is really whether or not these frogs can become reproductively mature themselves. And so, again, this is not every example, but just some examples. We've got on the left there, these are Lithobates sevosa from Cecilia's lab, fertilized from cryopreserved sperm, and Axyrus boreas in the middle there, and the Latoria aurea on the right there is from my lab, which was published in Natalie's special edition as well. So just to finish off this section, we thought we'd give you sort of an example of how a real protocol was developed in Cecilia's lab. And so they started off with spermic urine, non-lethally collected, and they extended it 1-to-1 with their selected cryoprotectant, which ended up being 10% dimethylformamide, or DMFA, with 10% trehalose. So often people will use a sugar or a saccharide in the cryoprotectant to help manipulate the osmolality, which can be very important for how the cells freeze. So in this case, they used the styrofoam box method, and you can see they had a rack that they had fashioned that held the straws in place and was suspended exactly 10 centimeters above the liquid nitrogen level for 10 minutes, and then stored in liquid nitrogen afterwards with rapid thawing in a water bath. And this is an example of a cooling rate that you can generate using your thermocouples. I think we'll see another one from Beck as well. So just to finish off, Cecilia's put together a really nice timeline. It's not exhaustive, but it's just some of the milestones that we've had over the years. So one of the first studies with cryopreservation for Bufo americanus was in 1972 with Barton and Gutman using ethylene glycol. And then in the late 1990s, we had the first successful post-thaw fertilization from cryopreserved sperm. This was from cane toads in Australia, where they're a fantastic model because they're so invasive. In 2005, there was first report of post-thaw motility and fertilization in Xenopus laevis and tropicalis. And the interesting thing about this protocol is that it actually uses egg yolk and sucrose and doesn't have a penetrative cryoprotectant, which is quite unusual. In 2011, there was the first publication for post-thaw sperm motility and fertilization from live animals with non-lethal sperm collection. So this was quite a cool paper out of Russia. In 2013, Langhorne et al. Had the first endangered amphibian species with cryopreserved sperm creating offspring. And then between 2014 to just recently, there's been quite a bit of work on different tree frogs in Australia, including an endangered tree frog.
Hello, everyone, and thank you for coming on this journey into amphibian cryopreservation with us today. I will be your guide into caudate cryopreservation considerations. I am not going to go into the step-by-step process of how to freeze caudate sperm because that was very well covered by Dr. Langhorne. Currently, the process for freezing caudate sperm is very much the same as it is for anurans. As we move forward, that may change in time. There's a lot we still have to learn about salamanders, and we're still in early stages of assisted reproductive technologies for caudates. But as it stands, the current process of freezing caudate sperm is a very similar process to anurans. I will touch on it, but I won't go step by step. What I want to focus on today is the difference between caudate sperm and anuran sperm and how to kind of think about it and handle it when you're doing cryopreservation in these species. So the first thing to consider when you are freezing caudate sperm is that caudate sperm is very large. This can vary, of course, species by species. Some species do have smaller sperm or sperm that is about the size of anuran sperm, but in general, they're very big. And this can be up to between 500 and 800 micrometers. That's about over half a millimeter, and that means it can be seen with the naked eye. This is important because sperm that big is very easily damaged during the freezing process. Of course, all sperm is susceptible to damage during the freezing process because of the formation of crystals and the chemicals we're exposing them to. Being larger, they're more susceptible to that. But also, these large sperm are susceptible to just physical damage by you handling them and manipulating them and pulling them through the cryo straws. So just handling them very deliberately and slowly and gently is important. The next thing to remember and understand is we have both external and internal fertilizing salamanders. So if you call back to that intro lecture that I gave on ARTs and salamanders, there's 3 families of external fertilizing salamanders. Their sperm in general works very similarly to anuran sperm. That means it's triggered by the environment. Typically this is an osmolality change, so being triggered by by water. This is important to know if you are assessing the viability of sperm and using motility to do that. Motility isn't, if you're doing a scientific experiment, motility isn't always the best way to assess viability. I'll touch on that in a minute with the internal fertilizers. But if you're just doing a quick check to see, does this animal have sperm that is going to function? Just looking at a slide and seeing if the sperm is moving is a great way to test if the sperm is good. Doing that with the external fertilizing salamanders using an osmolality test by dripping some water on it is very useful. The internal fertilizers are quite a bit different. So if you recall, the internal fertilizers use spermatophores, which are either deposited in the environment or directly into the cloaca of the female during amplexus. You can use spermatophores and directly freeze them, and they can be pulled up into straws and frozen the same way as either the spermic urine of the anurans or the milt of the externally fertilizing salamanders. The other option, which is helpful, especially if you want to assess individual sperm and the viability, is to do what was discussed in the hormone lecture and collect milt, or rather a broken down spermatophore from these internal fertilizers. And that is done by giving them hormone and then massaging their testicles and collecting the sperm directly from their cloaca in a liquid form. The benefits of this liquid form, this milt or broken down spermatophore really, are it's easier to use in IVF, so you don't have to break down the gel of the spermatophore, and it's easier to assess viability. So in internal fertilizers, viability can be tricky to assess because the sperm is not typically active. The sperm activity is not driven by osmolality, so just dropping water or salt or many different chemicals that have been tried in many different trials on the sperm doesn't seem to drive activity. Sperm is triggered by contact with egg jelly. So, this makes sense if you think about how these internal fertilizers work. The female picks up the spermatophore, she tucks it into a little pocket in her cloaca, and then as the eggs pass through the cloaca, the sperm is directly deposited onto the eggs. So the sperm would be wasting energy if they were moving around before touching the eggs. So I'm going to show these videos here. You sometimes do see motility before adding egg jelly, but it's not quite the same. So this first video is a spermatophore, and you can see just a little minute amount of wiggling. And you do get wiggling in the sperm if there has been any kind of manipulation of it. So this next video is of some milt or a broken down spermatophore that was collected from an animal. And you tend to see some slow motility in this. Now this is a very motile sample. It's typically not this motile, but you see this slow swirling of the sperm, and that's driven by an undulating membrane on the tail. It's not like a whipping action or anything like that. It's just kind of slowly driving itself around. Now if you add egg jelly, you get a much faster movement. It's still spinning right here. There are other enzymes that will, if it was close to the egg, that would cause the actually propelling of the tail, but it will move much faster if egg jelly is involved. So, the point here is using motility to do a quick assessment of viability is not a really good measure in the internal fertilizers. If you want to truly get a good measure of viability in the internal fertilizers, or if you're doing a study and you're measuring viability, staining is going to be the best option. The safest stain we've found in salamanders so far is the trypan blue and the Hoche staining, but Cyber 14 and the propydium iodide, they work too. It's just the propydium iodide seems to be toxic to the sperm and kills it, and that kind of ruins your viability test. So the trypan blue and the Hoche staining seems to be the better option. So as far as the cryopreservation process, for salamanders, the recommended cryoprotectants are DMSO And my personal preference is to use DMSO and BSA. So DMSO is dimethyl sulfoxide, and BSA is bovine serum albumin. And the BSA just seems to really add to the survival rate of the sperm and give a little extra protection. Now that's when, you know, you're using the milt that is very protective of those individual sperm. For spermatophores, spermatophores seem extremely resilient. There are studies out there that show you can just freeze them in spring water. I do recommend using a cryoprotectant, but you don't need to use such a toxic one. So sucrose is a very good cryoprotectant that we're seeing right now for spermatophores. As I mentioned earlier, the process of freezing is very much the same as for anurans. So you use the cryo straws and you slowly lower that into liquid nitrogen from 10 centimeters above the liquid line. You can thaw either fast or slow, either in the hot water or at room temperature. My personal preference is the room temperature because I saw less breakage, but that seems to be contradictory to other people's results. And, you know, both seem to get the same rate of return on viable sperm. So I don't know that there's a big difference there. There's still a lot we need to learn and still a lot that needs to be refined with cryopreservation protocols. The protocols we have right now work, and that's great. I mean, we've been able to successfully get second and third generations of cryopreserved babies, but at the same time, there's always the possibility for improvement. We've, as I mentioned, based these protocols off of anurans. You know, I've just been talking about how different caudate sperm is from anuran sperm, and that might be something we take into consideration in how we freeze it. While this works, it might be better for the sperm, or even easier on us, to try and pellet the sperm and put it over dry ice. That might be especially efficient for spermatophores rather than sucking them up into straws and trying to get them out. It might be easier to put them in tubes and pellet them over dry ice. So those are just recommendations for people, all of us, to try as we move forward. Here are some resources that are, you know, discussing all of what I've discussed above. And then please, please, please feel free to contact me if you have any questions. I'm always happy to talk about salamanders and assisted reproduction. Thank you very, very much.
So we've had some nice discussion already about the kinds of techniques that are available and the progress that has happened so far. So I thought we would just perhaps wrap this seminar up by talking about a real life sort of integration of this as an applied technology into some programs here at Taronga. Next slide, please. Oh, there we go. So strategic wildlife banking is certainly a program in applied research here at Taronga across a variety of different taxa. We have biosecure manned banks and alarmed banks, both at the Sydney campus at Taronga Zoo and also Taronga Western Plains Zoo, which is about 500 kilometres inland of the coast, just directly west. And we're using this as one component of an integrated strategy. So on staff, we have disease and health experts, we have captive rearing experts, particularly in the area of amphibians. Mr. Michael McFadden is in charge of that breeding programs here, as well as both Justine and I as reproductive biologists, and Dr. John Daly, who's just come on board here at Taronga. And as Rose mentioned earlier, he's actually been part of developments in the cryopreservation of coral larvae and has had some experience in fish species as well. So this isn't necessarily, I think it's important to point out, this isn't necessarily going to be an applied technology that's applicable to every single species that you work with. And it's important to have prioritization tools to be able to identify the key species that are going to benefit from the addition of this technology into their breeding programs. But it's certainly something that's a little bit easier perhaps to do in those species that external fertilizers and also produce a lot of offspring in one clutch. And obviously our ultimate goal in all of this is to build reserves of genetically diverse individuals across taxa for the integration into conservation breeding strategies and breeding for release programs. So getting some of these animals out into the wild. Next slide, please. So it's a really good time at the moment to be looking at amphibian biobanking. And as many of you on this call have probably contributed to, the 2007 publication of the Amphibian Conservation Action Plan highlighted that genome resource banking is a logical next step in how to, I guess, upgrade your conservation breeding programs. And as Rose and Cecilia have highlighted in the early parts of this talk, there's really been some significant research and development over the last decade, particularly around those non-lethal methods and across the board, as you'll find out in this seminar series. And some of you have contributed to the 2022 publication of the new ACAP, where genome resource banking is going to be dedicated a whole chapter in that publication. Here more locally, the Australian federal government has also identified genome resource banking as a really integral part of captive breeding programs and the development and the investment in those in this country over this decade. So that's put us in a pretty good position here at Taronga Conservation Society. We already have well-established amphibian breed for release programs, and we've collaborated with others in the development of some of the technologies necessary, excuse me, in the lead up to applying cryopreservation specifically. And we have, obviously, as I mentioned, the 2 cryodiversity banks that have been established since the mid-90s, and certainly over the last 5 to 10 years has expanded. Next slide, please. So a couple of years ago now, in the aftermath of the Black Summer bushfires here in Australia, there was renewed interest in biobanking as a strategy to preserve a number of species after we saw a drastic loss of millions of individuals across many taxa. So the goal of this specific line of research is to establish this strategic banking approach to safeguard as much genetic variation as possible in living frozen sperm biorepositories of key breeding programs that we already have established at both Taronga Zoo, but also Zoos Victoria. Now we're looking at next slide, next part. So our approach is this simultaneous refinement of these sperm collection and cryopreservation techniques, as well as being integrated into these natural breeding processes for these species. We have university partners at the University of Wollongong that have done extensive research on the spermiation protocols for Latoria burlongensis up the top there, on the left specifically, and also with the corroboree frog, the little black and yellow frog there, which is a pseudofren species, as well as partnering with the University of Newcastle. And as Rose mentioned, they've done a lot of work in Latoria species, and that's what we're basing some of our testing on when we apply to these species, Latoria burlongensis, Latoria castanea, and the spotted tree frog, which is held down in Victoria. Next slide, please. So some of this is, for us, time sensitive for this cryobanking. The animals that we have in our collection, particularly for the Boreal frog, they in the wild typically live 3 to 4 years, and we've already had some of these individuals who were collected directly from the wild 2 years ago. So they are certainly aging, and we've been able to collect samples from them over the last 2 breeding seasons, and we're freezing or cryopreserving those samples with the best practice we have at the time. We've been comparing Rose's method with the DMSO sucrose and a programmable slower rate freezing method with Cecilia Langhorne's slightly different penetrating and non-penetrating combination of DMF and Trailose with a faster freezing rate, which we have tried to make a little more field friendly by using a dry shipper. Now, obviously, part of the consideration that you have to have, and I'm not sure if this was spoken about in the spermiation and hormone seminar that has happened already, but we're integrating within a natural breeding program. So we don't have access to all the animals all the time, but as an example with the Borealensis here in the picture, they typically, their breeding season runs from September through December, but the majority of natural breeding is going to occur around October and November. But we have been able to collect really good quality samples from our males after they've stopped calling into December and into January. So we're able to collect after they've been used for natural breeding and produced offspring that way. We also tend not to, or haven't, stimulated those animals with hormone within 4 weeks of a natural breeding or of another hormone induction protocol. So we try to collect sperm more than once every season because they do produce very small amounts. And again, that's another reason why we use straws versus cryovials, is because it is low volume. With the corroboree frogs, they have even smaller volume, and they also produce much lower concentrations. So there are other things you need to keep in mind when you're looking at freezing these. And we collect from those corroboree frogs over many hours and pool all of that together. So we're looking at collecting for 24 hours from those animals every couple of hours. What we're, as I mentioned, looking at is which is most, the cryodiluent and the dilution rate most applicable to the species we're working with, the freezing rate, but also importantly, that thawing rate that a couple of our speakers have talked about already. So these are ongoing, and unfortunately I don't really have a lot of results I can show you today, but I'll do that in the next slide. And as mentioned again in one of the other talks, and here again, was looking at trying to optimize those assessment measures. And we've certainly seen with the corroboree frog, they're very different to the Latoria species in terms of their motility, the rate and the directionality of that motility. So we've been doing these trials and also genetic analysis of these individuals at Taronga Institute of Science and Learning. It's important to have that genetic analysis in terms of management and trying to get a broad and evenly spread biobank across the individuals that are represented. And as I mentioned, and others have mentioned, looking at field-friendly and cost-efficient methods for sperm banking, and that's something that's also going to be covered in subsequent lectures. But we have tried with our initial experiments in the Boreal and the yellow-spotted frog to adapt those to being field-friendly. So you can just see here in the table that we've thought out a few for our preliminary results, and you can see for the Borealensis at the top that we're still retaining between 50 to 60% of the total motility with a reasonably good progressive motility. And this is following activation through an osmotic change, placing that in fresh water, and a post-thaw live percentage of cells retained at about 50%. And so this is a drop of almost half of that initial motility, and also post-thaw membrane intact. So that's another thing you have to take into consideration when you're going to use these samples in the future, as to what your effective concentration is actually going to be within your sample. You can see that the Latoria castanea didn't have quite as good post-thaw survival and motility, but will be, obviously that's just one individual, and we'll be looking at evaluating that more closely in coming experiments. And you can see here on the left, we're actually collecting spermic urine, and that those protocols were developed by the University of Wollongong, Dr. Amy Siller, who you'll hear about in this seminar series as well. Next slide, please. So this is looking at trying to match that freezing rate that Cecilia Langhorne has established using liquid nitrogen, which is obviously very difficult to take into the field, especially if you're out there for multiple days. And it does seem to approximate the freezing rate, though you can see the range that we have is quite broad, between -17 to -24 degrees per minute. So this might benefit from something that's called seeding, where you can artificially stimulate the formation of or the nucleation points within that straw or vessel. And that can be as simple as giving it a quick flick at a specific point or specific temperature. So we might be looking at experimenting with that too, to try and get it a little bit more standardized. Next, please. So this is just to give you a visual representation of the method that we're using. So our animals are actually in biosecure facilities, so we can't bring the animals to the lab. So we collect the samples into an equitainer, which can hold those samples at 4 degrees, and we leave that equitainer down in the biosecure facility until we've finished all of our collections. We pull them at the time of collection, and then we'll bring them up to the lab for assessment when we're done. We're just using a simple camping fridge freezer that you can get from any camping store, and that's to process the sperm samples and to keep them cool. We add cryoprotectant at 4 degrees, and we also load and seal the straws. We have pre-barcoded straw labels, which I'll show you in the next slide. And again, this is just looking at the dry shipper method, which has already been standardized in a paper by Justine and Terry Roth in 2004, mammalian sperm freezing. And we're using the ultra rapid thaw method of 5 seconds at 40 degrees, which approximates about 2000 degrees a minute. So we're the next steps for this program is to continue that development in the remaining species. We've obviously done a lot in the Boreal, and the season for the northern and southern corroboree frogs is upon us now, so we'll be doing that. And also it's important to, when you're thinking about the packaging that you're going to be using for your sperm within your biobank, is to conduct those IVF trials to know what concentration of sperm you might need in order to get successful fertilization. So essentially, how many sperm per egg, and what are the clutch sizes that you'll be dealing with in the species of concern. So we'll be working on that in the coming years, as well as looking at translocations. Our captive breeding programs already have translocation within them, and getting the permissions to put some of these animals out into the wild. Next slide, please. So just finally, making a point about the quality of the data collection. The biobank you have is only going to be as good as the metadata and data that go along with it. So it's really important that you keep quality, consistent records for each of your collection attempts and cryopreservation. So this is just an example on the left bottom of what we've now moved to in the last couple of years for the labeling of our straws, and that's using a barcode printer and a sticky label. This is just represents the date and the individual identifier for that animal and its house name there in brackets. I always keep a copy of the exact label, and we use just the same point and shoot data readers that you might find at a supermarket. It's nothing super special, and it's nothing particularly expensive, which is also important for that. And it can connect directly to Excel for data input or checking. So you can see here some of the data that we're currently collecting, at least around the cryopreservation part, is looking at keeping records on the cryoprotectant, how it was added, the volume in which your straw is filled, the timing of that, and the method that you used. And obviously there will then be tracking data in your biobank as to where those samples are located. There's additional metadata, obviously, around those animals that's going to be kept with the animal records that you need to be able to relate. And we'll certainly be working with the rest of specific biobanking community for the IUCN biobank specialist group, as well as the amphibian specialist group, in trying to figure out what's the most important metadata that we need to keep tracking and updating to correlate with these samples in order to make sure that they can be most effective in their use. And that's it for me. Thanks.